Hi, this is Dr. Rob Morgan, the Chief Medical Officer for Let's Get Checked. And today we are excited to have join us one of our brilliant in-house talents, Dr. Gwen Murphy, who is the Director of Epidemiology and Clinical Studies for Let's Get Checked. So hi, Gwen. Thanks for finding some time in your day.
Pleasure, Rob. It's good to see you.
So let's jump right in. You're the Director of Epidemiology. What the heck does that mean? What's an epidemiologist? Tell us a little about what you do. And I think also it may be interesting for people to hear your career path as to how you found your way to where you are today.
Sure. Thank you, Rob. So yeah, what is an epidemiologist, right? Even my own family a few months ago didn't really have a good grasp of what an epidemiologist does every day, but now it's become part of the mainstream for all of us, which is great for me. It means that a lot of people are interested in what we do and what I do. So epidemiology is the study and the analysis of distribution of health and disease in a population. So the who, the when, and the where of health and disease. And so for me, I've had a pretty varied career path to get to epidemiology. I started off as a molecular researcher, molecular biologist, I guess. And I had an undergraduate degree in physiology and then realized that I really wanted to pursue physiology more and get really into the weeds a bit more, and decided I was going to do a PhD. So I took on a PhD in medicine, which I did at one of the hospitals here in Dublin. It was a Trinity College hospital. And it was all gastrointestinal disease focused. I thought the gut was pretty fascinating. I still think the gut is pretty fascinating. Because in high school biology, our biology teacher used to say that while the food is in your gut, it is not in your body. And I just thought that it was like mind-blowing to me that it's an external environment, but it's in your body. That just blew my mind. So I put a few years into studying that. And I did a PhD, which was really focused in inflammation and gastrointestinal cancer. And then as a result of that, I discovered that the bits that I liked, I was in the lab all fingers and thumbs. I was not good at pipetting. I was not good at carrying. So I would spend 2 days staining a piece of tissue, and at the last moment I would wipe the tissue off the slide. I was not suitable in the lab. So I decided what I really wanted to do was look at the data that was being produced and study that data. And so I looked for a path to do that. And I wound up I was a cancer prevention scholar at the National Cancer Institute in the US. So I was one of the first Irish participants which was admitted to that program. And as part of the program, what they do there, it's a fabulous fellowship. And what they do is that they pick people from all sorts of different disciplines. So an oncologist or a linguist or a psychologist or a basic lab scientist, and they give us all a master's in public health. So a basic grounding in public health and epidemiology so that we can all speak to each other at the same level. And then they put us all together through a fellowship program. And so I went to the US. I lived in Bethesda near you, Rob, and I studied at the NCI for a period of 2-3 years. And then I wound up staying on as an epidemiologist there for 10-12 years at the National Cancer Institute, studying, again, gastrointestinal cancers, stomach cancer, esophageal cancer, colon cancer as well. And then I moved back to Dublin and took up a position at Let's Get Checked. Again, we offer lots of home testing at Let's Get Checked, and cancer prevention and cancer screening form some of those tests. So I felt like that was a great opportunity to further explore and put a kind of a different perspective on what epidemiology has meant to me. So that's kind of a long-winded answer, Rob, sorry.
No, it's a fascinating journey, and obviously one that, I'd say you need to go to school, Gwen. You need to learn something along the way. Make something of yourself. Very impressive career path, certainly, and a lot of hours, a lot of days, a lot of years invested to get where you are. But we're certainly so fortunate to have you as part of our Let's Get Checked team, because as you know, we have, all of us at Let's Get Checked have seen such a seismic change in many ways to where we are today versus where we thought we'd be today. If you go back to even as recently as when we last saw each other in person in Dublin in January, no one could imagine we would be where we are today, as a society, as a country, or countries, certainly as a company. And so, you know, talk a little bit about what COVID-19 has kind of meant to you and to your role at Let's Get Checked. I mean, obviously, we're a home diagnostics company. We do a lot of different testing. You mentioned some of the cancer screening that we can offer at a home diagnostics level. We know that we also do, you know, testing for lipids and thyroid and sexually transmitted disease screens. But COVID has just turned everything on its head. So can you talk a little bit about what the process has meant for you at Let's Get Checked, most specifically to COVID-19?
Sure. So as I said, you know, I joined Let's Get Checked last year, and my focus was really on chronic disease and prevention of chronic disease. And COVID was interesting. It popped on the radar, obviously, kind of the end of last year, that they were seeing a lot of influenza-like illnesses in China. And then as December wore into January, it became clear for me as an epidemiologist that this was reaching into the mainstream. This was no longer some sort of niche thing that a bunch of epidemiologists were watching. This was something that was going to affect all of us. And it became really clear by the spread of the disease that something unusual was going on. And so epidemiology really seeped into the mainstream at that point. And I think at Let's Get Checked, we realized very quickly that we had developed as a company over the last 5 years to produce a whole end-to-end testing platform, so that we control, obviously, the production of the kit, the delivery of the kit, you know, somebody taking the kit and informing them how to do that, and the best way, the best kit with the best performance at home, and then at the back end then delivering results to patients. And we realized very quickly that we could do that for COVID, that we were producing essentially a very quarantine-friendly test. So we decided very quickly that we needed to get involved. We needed to spin up very quickly a test that could really help people out in a time of complete crisis. And so the first test that we ran for COVID was a swab test for healthcare workers, for essential workers who were frontline and really needed access to testing. And at that point, and probably still, there's a real bottleneck around testing. Testing is the crux of how we control this pandemic. And at that point, when we were starting in that COVID testing sphere ourselves, we realized that we couldn't do enough to open up access. And so we started with healthcare administered tests and realized that we needed to move through FDA approval to get tests into people's homes, so that we could deliver tests right to the people who might be isolating at home, who might be quarantining at home. And so we could reach them in a way that would mean they didn't have to go to the supermarket and queue up for tests at testing sites or at clinics, or we could reach them directly in a way that was probably most efficacious for preventing infection.
And so I want to get into the weeds here a little bit, and I apologize dragging you into your epidemiology, 101 for the rest of us here. But I know that there's talk around this idea of R0 and what the infection rate of COVID is, and I'm hoping that you might explain to us a little bit about why it's important that we not only, be concerned about COVID-19. We're concerned about it because society is concerned about it, but maybe some of the more nuanced reasons about how infectious this virus really is when you sort of put it in comparison to some of the other more commonly known viruses, and a little bit around that. And again, kind of highlighting why, if you can do a test on your own from the safety and sanctity of your own home, why that really is a crucial piece to the battle here.
Yeah, so the R0 is, again, it's an epidemiologic term that we've all become very familiar with over the past few months. And so what we're describing is how infectious is that disease relative to seasonal flu. So one of the things that we learned with coronavirus was that if you were a case of COVID-19, you were more likely to infect 2 or 3 people around you who could potentially pick up that virus, right? For seasonal flu, that's like 1 or 2. So it's more than seasonal flu. It's less than measles. So measles has a really high R naught. You could infect maybe 10 people around you with that. And so what's important there is that if I'm diagnosed as a case and if I'm quarantining at home, I should be contact traced, right? So that my public health department is going to come and ask me, how many people were you in contact with? Because they then have to go and test all of those people around me, because now I'm the index case. I'm the person of interest here that needs to be explored. And so knowing how the density of how many people I'm likely to have infected is really important. One of the great things about epidemiology is that we learned as well that we can fight this infection with some basic principles of public health and personal hygiene, right? We learned that this is a respiratory virus. It's passing in aerosols from person to person. So if I'm speaking very close to you, you could pick up one of those aerosols. Maybe you rub your eyes, maybe you put your hand in your mouth, we all touch our face, so we're spreading potentially virus particles from our hands or our surfaces that we're touching into our epithelial surfaces, and that's how people get infected. So what we're learning, or what we learned very quickly with this respiratory virus, was that we can drastically reduce the chance of somebody getting infected by social distancing, by keeping back when you're interacting with somebody who's not from your household, by wearing a mask when you're outside, by washing your hands frequently, sneezing into your elbows. And what those measures can do is drastically reduce the R naught. So the R naught is not a concrete term. All things being equal, we know R naught is generally about 2 to 3 for COVID. It's less for seasonal flu, it's more for measles. But the R naught, we can move that. Every single person here has an opportunity to modulate their own risk for infection, right? We can control the R naught for COVID-19 by social distancing. And so it's a really interesting idea that these basic public health principles, basic hygiene practices can be the most effective tool that we have against COVID-19.
I'm really curious to see what the impact may be on all of the other common disease, viral infectious states that we otherwise would normally see. So, are we going to see a lower incidence of influenza, the common flu this season, because of some of this? I think it's going to be, this is the time to be an epidemiologist. You guys are going to have enough data to look at for the next decade to keep lots of PhD programs busy, I suspect.
Yeah, I like to think of this as the launch of a whole generation of epidemiologists and public health practitioners and statisticians and mathematicians. And there's a quote from, I don't know if you ever watched Buffy the Vampire Slayer, Rob, but there was a great quote from
Not
the
that
program.
I'll ever admit.
There's a great program about, every girl who could be a slayer would be a slayer. I like to think that everybody who could be an epidemiologist will be an epidemiologist, that all of these high school kids and kindergartners right now who are interested in numbers and interested in counting, or maybe just detectives, they like to solve puzzles. Maybe we'll launch a whole generation of epidemiologists. And I think this is the first global pandemic in our lifetime. It probably will not be the last. And certainly, there's lots of countries around the world where epidemiology and epidemics, that's their bread and butter, right? Whether it's cholera or whether it's yellow fever or dengue fever, there's all sorts of diseases around the world that we look to public health practitioners to control. And so this is the first time epidemiology, I think, has been right out front and center. But I'm hoping that that it has grabbed the public attention and that people feel intrigued enough to want to pursue it as a career path or as a passion. We're seeing already is that flu numbers seem to be down, certainly in the southern hemisphere already. We're seeing that these measures, as you would suspect, if you're trying to prevent one respiratory virus, you're probably also going to have a knock-on impact for controlling another respiratory virus. And the news is that does seem to be the case, is that the flu numbers appear to be down. And also because certain members of the population who might be more vulnerable to flu and to the serious consequences of flu, because, you know, it's important for all of us to remember, kills people every single year. And so we probably are saving lives as a result of protecting people from the flu, as well as protecting people from the serious consequences of COVID-19. So yeah, there should be knock-on effects. Also, you know, for all of us, I have young kids. I don't know how many GI bugs I get every winter from, you know, frequent hand washing is good for all sorts of reasons. There should be impact for lots of different contagious diseases, hopefully.
Yeah, no, I think it's going to be interesting to see, even as we know at some point this pandemic will be brought under control through the efforts that you mentioned, and, you know, hopefully a vaccine at some point and some of these things. But hopefully there's going to be a lot of long-lasting, you know, lessons learned and behavior modifications.
Yeah.
Well, I do think the epidemiologists had better get their rest, because I'll tell you, I think you guys are going to be really front and center quickly. Hopefully quickly. I believe that this whole issue around antibodies and the significance of antibodies is going to take center stage if and when, I won't say if, but I'll say when we have vaccine options out there. Because I think that you and I understand that the way a vaccine works is you're essentially triggering the body's immune system to build antibodies, to build protection against the virus that you're trying to fight. And so when a vaccine or vaccines become available, we're going to need to be able to measure what that individual's body is doing as a response to the vaccine, and then sorting that out in the context of, is there a break point in terms of that immune response above which now you've got immunity and below which you don't. And I think about some other vaccine programs over the years where, you know, not with COVID obviously, but with other disease and viral disease states where it's important not just to get the vaccine, but you then have to have your your antibody titers checked, meaning looking at your antibody levels to figure out, are you in the clear, are you safe, or are you not? So I think it's not going to be as straightforward as just a binary, you got the vaccine, you're protected. It may be, and that would be great if it was, you know, one size fits all on the vaccine, but more likely it seems that there will be a certain amount of measuring of antibody response that's going to have to be factored in. So, you know, stay tuned, right?
Yeah, absolutely. And the type of antibody, like you say, there's so many different types of antibodies that we produce, and we have a whole host of an immune response, not just antibody production or not. There's a whole host of other cellular immunity that's involved. And so measuring, you know, what they'll be doing as part of the vaccine trials is classifying how people respond to the vaccine and seeing how their whole pattern of antibody response that comes back from those. And I would say, you know, you and I know that we're here today because of the success of vaccination programs, right? Vaccines make adults. There is no better and more efficacious public health than vaccination. It saves millions of lives. So we're all super hopeful that the vaccines will be released soon, and that the trials will be completed quickly and show that the vaccines are efficacious.
Yeah, no, I completely agree. So a lot to come there. Hopefully a lot to come there, right? We hope that, you know, the conversations will pivot in the coming months to some of these vaccines and being able to measure efficacy and safety, and then how do we roll those out at large population levels. There's so much work to be done there, but I know a lot of it is ongoing at a very, 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 very fast pace, a pace that's faster than any of us have ever
been seen before. Absolutely.
Yeah. Well, and on that topic, I do want to pivot a little bit. I appreciate your you know, your sort of perspective around the various tests, but I do want to talk a little bit more about Let's Get Checked test, the PCR test. You mentioned the lower nasal. But the thing I wanted to ask you about, because you really were the point person on this at Let's Get Checked, is the whole process that you went through and brought our test through with the FDA. So one of the things that we're obviously very proud about and has allowed us to expand the testing around COVID as a company has been the fact that we got FDA approval, or FDA authorization, correctly stated,
Yep.
For our test to be self-collected for home use, meaning that they didn't have to be done by a physician or by a healthcare professional, in an office or in a hospital or in a clinic. One could literally do our PCR test, or at least collect their swab for the PCR test at home. And I know that you burn the candle at all ends 7 days a week to make that happen with the FDA. And I'd be curious to hear a little bit more. I know a lot about it, but I think others might be interested to hear what that process was like, what your experience was like with the FDA, and what it means to get that type of authorization.
Sure. So I think there's a number of things when we're thinking about testing for a lay person in particular. What we need to bear in mind is that there's two components, right, to the test. So there's the lab test that we do in terms of PCR to determine whether or not there's virus present. So we had that identified and pinned down very quickly, that we knew the type of assay. We chose an assay that's got a particularly low limit of detection, so it can detect a very small number of viral particles. If it's in your nose, we will probably be able to detect it on the swab, right? And so we picked an assay that we thought would work for us because of its extreme sensitivity, and because we could be very certain that the assay would be specific to SARS-CoV-2, right? So specificity is important as well as sensitivity. The other part then...
And our assay,
yeah.
just to interrupt real quick, sorry, because it does come up a lot, is, so when people say, well, let's get checked, you're doing this COVID-19 testing, what's your sensitivity and what's your specificity? How do you answer that one? I think you just did, but I'll give you the question clearly so you can answer it clearly.
Sure. So our sensitivity is up at 98, 99%, and the specificity is really closer to 100% in working terms. So our questions around the assay performance, and I will say a lot of the PCR tests, they're very robust, they're very sensitive, they're very specific. So, we did a great job, I think, as a scientific community in moving at breakneck speed from the genetic sequencing the genome of the virus to producing tests at a large scale that could measure that virus in URI. I think that's never been done before, and that was a huge leap scientifically. Obviously, with our experience as a test company, Rob, for the last 5 years, we know our customers, we know the points around where they need extra help or support with using a test in their own home. And so we wanted to be safe and to be sure that people can use our test and give us a proper sample that's going to be able to be measured in the lab using our PCR test. So we spent a lot of time working on what that test should look like and what the supports should look like. We made supportive videos. We've produced some great instructions for use to make it as easy as possible for people to take a great sample and send that back to our labs then with some ease and be able to receive the results. Ideally performing equivalently to what you would get in your doctor's office or in a hospital. And so that's what we've come up with, and that was what we worked through the FDA for authorization. They looked at all of our data. They looked at our end-to-end, because we as a company are responsible for the kit, the whole kit that arrives, the manufacturing of the kit itself, as well as that arriving into your home and what you do with it in your home, as well as the results, as well as pushing the results to your app. And so the FDA, it was a unique review, I think, in that regard, because they reviewed us end to end. They reviewed our entire process. And they found that they were happy with it and they were ready to authorize it for release on the market. So it was a great process to be able to work back and forth with them and to have their input on what we should be doing.
Bumping up on time here, but I'll tell you something.
Sure.
I'm going to throw one big sort of monkey wrench at you. I know this is a hot topic right now in the COVID testing world. And so I'd like to just get your sort of hot take on it, if you will. And that is around this idea of saliva testing. The test that we offer for Let's Get Checked currently is this lower nasal swab, which means, essentially a Q-tip-like swab in the lower part of the nose, rubbed on the inner surface of the nose. That's our collection for COVID-19. That's our test, the PCR test. Is a fair amount in the media right now around saliva testing
Mm-hmm.
for COVID-19.
Mm-hmm.
And I know that one of the duties and responsibilities and commitments that you have at Let's Get Checked is to really keep your finger on the pulse of really all testing that's out there, making sure that at Let's Get Checked that we can be on the front end of all of that, that we can pivot to anything that is better, you know, in a very general sense. If there is a better test out there, a better mousetrap, so to speak,
Yeah.
that we are able to offer that as well. But a lot about saliva right now in the lay press, and so I just love to get your thoughts on that. I know you've looked at this quite closely, continue to monitor it, but where are we with saliva testing? Do you think that's something that we're going to pivot to at Let's Get Checked, and why not if the answer is no?
So for us, I think what was most important for us was the sensitivity and the performance of the test. We wanted to ensure that whatever test we give you to do in your own home, it will perform as well as the test that you would do in your doctor's office or in the hospital. So that equivalency was really important to us. And we felt that nasal swabs, you know, based on the data available, seemed to be the best way to get that equivalency in performance. Saliva seems like an easy test, right? You just spit in a cup and you send it away. What we're seeing is that there's a couple of complications with saliva, that it's a difficult specimen type to work with, and I think FDA have said the same themselves in some of their most recent correspondence and town hall meetings and things like that, that it's tricky to work with. There's some stability issues. So saliva may work okay if you're doing it right there and then, and it's going immediately onto an analyzer. What the problem with saliva is if there's any kind of delivery that's required, if you're taking your saliva test at home and going to the lab, that sample might not be as good by the time it gets to the lab as it is when you spit into the cup in your own home. And so the stability concerns were probably our biggest concern around saliva testing. And what you've seen in some of the saliva tests that are available is that you see this trade-off for, yes, it's easy to do, but then there's a decrease in sensitivity associated with it. And certainly we've seen that with some of the most recent saliva tests that have been approved. There's a compromise that you need to make if you're going to not have an extraction procedure for that before the test goes on the analyzer, the sample goes on the analyzer, then you're going to see a trade-off in sensitivity. And I think, unfortunately, that's what we see for saliva. In terms of worldwide, we need all tests. We need different tests for different situations. But for our own situation, we wanted to be very sure that we're guaranteeing a test that's highly sensitive and that it's working equivalently to the one that you would do in your doctor's office. So no, at this point, we're not ready to offer saliva as a sample type.
So if I'm understanding it, so just if I'm understanding it correctly, maybe stated a slightly different way, it's not that the saliva test is horrible or is bad, but it just may not be, it doesn't appear to be as sensitive, as specific, as good, if you will, than the swab PCR. The other thing that I sort of have bumped into, and you and I have been on a lot of conversations around this over the last weeks and months, is that the idea that saliva is easy somehow implies that the swab is somehow hard and difficult. And I would just highlight that the swab, the lower nasal swab that we're talking about, not the deep nasopharyngeal, not the deep oropharyngeal, but our lower nasal swab, the Q-tip in the lower part of the nostril, isn't hard to perform. It isn't uncomfortable. I mean, it's quite straightforward. So I'm not sure, you know, that it's sort of almost, the saliva is almost sort of addressing an issue that doesn't exist in many ways in my mind.
I think so that, you know, the saliva, literally it's taking a Q-tip. You know, my 6-year-old can do it. You take a Q-tip and you just put it in the bottom of your nose. Very easy to do. Not that deep nasal swab. Certainly you would need a doctor to do that. But for our home test, it's just the bottom of the nose with the Q-tip kind of swab. And that's as easy to perform. And we felt, well, if it's as easy to do that as saliva, we're going to go with the test that performs better and that has higher sensitivity and seems to have better clinical performance. The chances are, if you're very sick, if you've got tons of virus in your system, maybe it doesn't matter that the saliva test isn't performing as well. But we feel like we're doing a lot of screening, we're doing a lot of tests for people who think they're concerned that they might have been exposed or who have mild symptoms. And in those people, we want to be sure about their test will pick up the virus if it's there. So we want the sensitivity to be very high for those tests.
Got it. It's great. Well, I really appreciate the clarity there. Gwen, we're bumping up against our time. The good news is that there's so much stuff we didn't get to talk about today, which means that you're on the hook for another one of these, at least, maybe more. But I appreciate all your time, all your great insight, all the great work you're doing and continue to do. It's just been really impactful. And so thank you personally. Thank you for everything you've done. So we'll wrap there, and then we'll put another time on the calendar here, and we'll circle back, and we'll talk about all the other tests that we do, and,
sure.
and getting into some of the stuff that we can talk about something besides COVID next time.
Yeah, but, imagine
so much to
else.
talk about even here. Yeah. Gosh, we would not be nice. So thanks, Gwen.